stop.
Okay, we're live. Alright, so we have the heart. Again, we'll do this many times when we come back. It's okay, right?
I'm gonna get a good one.
We have the heart, and already we can see these flap-like structures are called the auricles. Right? They're associated with which chambers?
Atria.
The atria. Good. And then the larger pieces of the heart down here towards the apex, what chambers do we associate that with? So down here, the pointiest part is the apex, right? And the widest part is the
Base.
Good. Okay. Now, on the sheep heart, you do not need to worry about your sulci because they're covered with fat. Okay? All right. Now, also, you don't need to worry about the blood vessels that are entering or coming away because the four-legged animals have different presentation of their vessels. Here on your lab exam 3, those vessels will be tagged on the models of the heart.
Okay.
When I open up the heart, the first thing you want to do is, number 1, find the interventricular septum. And it doesn't matter which way the heart is turned or twisted, you should still be able to see the IV septum. Next thing you do is you compare the wall of the ventricular myocardium. Which side will have the thicker myocardium?
Left.
Okay. So if you know your IV septum, and you could see that this side is thicker than that side, then clearly all of this area here should be what side of the heart? If you know which side of the heart you are on, then you should also be able to quickly go through a short list of what structures would be found on that side of the heart. For example, which 2 valves should be found on the left side of the heart? The bicuspid, aka mitral, aka left AV valve. Are those associated with strings called chordae tendineae?
Mm-hmm.
Alright, so here we see flap-like structures with strings attached. See them here and here? Again, both sides were on the left side. So these are the chordae tendineae, and these flap-like structures here are parts of the leaflets from the left AV valve. Good?
Mm-hmm.
Okay. Notice these thick finger-like projections of muscle that they're anchored into. What are those called?
Capillary muscles.
Capillary muscles. Good. Now, we know that the blood has to be ejected from the left ventricle to the aorta, and blood can only flow from an area of high pressure to low. I'm going to flip this around. I'm still on the left side. Agreed?
Agreed.
Okay. But the exit for the aorta is clear up in the ceiling of the left ventricle. And I told you in lecture that when the ventricles contract, they push on the blood, and I used the water bottle as an example, remember that? And it pushes the blood superiorly, and these leaf-like valves will close, and the ventricles have to keep contracting to generate enough pressure to open these semilunar valves. Here's one pocket, and here's another pocket. See the cusp-like valves? You see that? Now, of course, we've sliced through one on this side. That's why we're only seeing one good one intact. But it is still coming off the left side, so that's why I know that's the aortic semilunar valve. Plus, there are no strings attached, so I know it has to be a semilunar valve, right? Okay. Now, I look down here in the left ventricle, and I see a lot of macroscopic muscle fibers, or macroscopic foldings of the muscle. What are these called down here? Trabeculae carneae. Now, in the atrium, I also see some macroscopic foldings, but those are called something different. Pectinate muscles, of course. Now on your lab exam 3, do not be myopic. Don't be so focused on the pin. Remember, the questions are important. If, for example, you see a pin right here, and it says, what layer of the heart is this? What would you say?
Myocardium.
Myocardium. But if there's a pin here, and it says, name this layer of the heart, what is it?
Endocardium.
Endocardium. It lines all the internal chambers and the valves. If you see a pin out here and it says, name this layer, epicardium. Good. Also, if you see a pin in this chamber up here, and it says, name this chamber, what are you going to say?
Atrium.
Left atrium. All right, so be mindful. Make sure you read the question, okay? Now, on this side of the heart, you can see a very smaller chamber, right? And we see our trabeculae carneae again down here. What chamber is this that my thumb is in?
Right atrium.
Nope, down here. It's ventricle.
Right.
This is all right ventricle. And I can see also, if I open this up a little bit more, this is called a moderator band. It is another kind of specialized cells that I told you about in class. It helps the conduction go to the left side of the heart. Not everyone has one. But if I open this up in here, I can see one of the leaflets for the right AV valve with the strings attached. So that's the right AV valve right there. See that? To get to the pulmonary semilunar valve, I need to close the heart, go to the anterior side of the heart. How do I know this is anterior? Let's get perspective again. This is the left, this is the right. So if I close it back like a clamshell, this must be the anterior side of the heart. And to get to the pulmonary trunk and see the pulmonary semilunar valve, I have to cut through this anterior wall of the myocardium. And if I do that, and open that slice up, I can see here's a cusp of the pulmonary semilunar valve. No strings attached, and I know it's a cusp-like valve, so it has to be a semilunar valve, but this is on the right side of the heart. Okay? And here's another auricle. And here is the right auricle for the right atrium. Did I get everything on your hit list? Did I miss something? I think I hit it all.
All good?
Yeah. Yeah, in order to get to the right atrium, I actually have to come up into here to get to the right atrium. Alright, so those are the structures that will be tagged on the sheep heart. We'll do this again for the human heart models, okay?